Yeah, good afternoon, dear students. So, hereby starting the new topic on acid-base balance and its disorders. Okay. So, previously discussed the carbonate metabolism in that. So, today I am just taking a new topic, acid-base balance and its disorder. First, the objectives of this topic is, first you should know about the normal blood pH, okay, and how does the pH which is maintained in the normal reference range. Suppose if it is not maintained because of various reasons, that is derangement in pH, that leads to a condition known as acid-base disorders. So you have to know about various acid-base disorders. At the same time, you should know how to interpret the ABG report in the above set conditions. Okay. So, in today's class, these are the main objectives of acid-base balance and disorder topic. In today's class, I am just taking. So, what are the normal reference range of pH, blood pH? How does the pH is maintained within that range? What are the various regulatory mechanism which will maintain the blood pH? So, I am discussing in detail about various buffer system, respiratory regulation and renal regulation. Okay. So, let us start. So, the pH of the arterial and venous blood, interstitial fluid, all this varies. So, you need to know about normal pH of an arterial blood. So, it varies from 7.35 to 7.45. Some books might mention 7.36 to 7.44. Okay. So, on an average, the pH of the blood is considered to be 7.4 is an average. The range varies from 7.35 to 7.45. Why you want to maintain the pH? Is that important? Yes, because in our body, the various activity, the metabolic activity, all these things which are controlled by the enzyme systems, they are all influenced by hydrogen ion concentration. So, you have learned that enzymes, they are specific in their action and their activity is mainly depends upon the pH of the surrounding medium. Okay. So, some enzymes will active at particular pH. So, like that, most of the enzymes which are active at optimum pH, that is neutral pH. Okay. So, if the pH is altered, that is influenced by hydrogen ion concentration, so that may affect the various enzyme activity in our body. So, what are the changes? Suppose if any hydrogen ion concentration changes, that bring about the altering the enzyme activity as well as all cell and body function get altered. Okay. So, and also it affects the uptake and release of oxygen from the hemoglobin. Okay. These are all basic important changes which occurs if there is any change in hydrogen ion concentration. So, that's why the pH of the blood should be maintained within the reference range, and if that is deranged, that will affect the various enzyme activity and also the following few functions get altered. So I already mentioned pH is this much reference range. So, do you think the acids and bases which are formed in the body? So, you might have observed in the laboratory, you see the various strong acids or bases, all those things, as well as weak acids and bases. Do you think these acids or bases are formed in the body? Yes. So, what are the examples for various acids or bases which are formed during various metabolic activity? Okay. So, I need to mention few of the acids and bases which are formed in the body. So, these acids, they are divided into fixed acids, they are non-volatile acids. And second one is volatile acids. They are washed out through the lungs in the form of example is carbonic acid. They are volatile acid. They are breathed out through the lungs. Okay. Fixed acids, example, phosphoric acid, sulfuric acid, pyruvic acid, lactic acid, and keto acids. These are the various acids which are formed during various metabolic process. Okay. That's why during various metabolic process, these acids get released to the bloodstream. It may alter the blood pH. Okay. At the simultaneously, even bases get released. I'll just show in the next slide the various example for the bases. Okay. So these are the few acids. It may be a fixed acid or volatile acids. So which get released to the bloodstream, which alters the blood pH. Okay. The bases, various metabolic, the sources. For example, so if you consume a fruit juice, okay, the citrate salts of fruit juices may contain bicarbonate salt. Okay. And deamination of amino acid, the removal of amino group from an amino acid that produces an ammonia. Okay. So this also forms a base. Okay. And also formation of this phosphate that also contribute to the alkali reserve. Okay. So these are the few bases, and just to need to mention, these are all which are formed through the diet or during metabolism. So they are formed, they contribute to the base sources of bases. Okay. So that's why during metabolic process, even acid can release, even bases can release, that may affects the blood pH. And suppose if that is too much deranged, how does in our body, which are able to regulate this and to bring down and bring the pH in a normal reference range, that you need to know. Okay. These are the various sources of acid and bases. So you need to learn about various regulatory mechanism which will maintain the normal blood pH. Okay. I think some of the things which you might have learned in physiology. So here, so you need to know the basic mechanism. Suppose if some amount of acids if added or some base is added, how does pH is maintained? Okay. So these are the various regulatory mechanism. One is the buffer system. There are three line of defense or three tier of defense to regulate the blood pH. One is, first line of defense is buffer system. Second one is respiratory mechanism. Third one is renal mechanism. So these are the three basic mechanisms which will regulate the blood pH. So the buffer system is the first line of defense which act immediately, and next is the second line of defense is respiratory mechanism, and third line of defense is renal mechanism. Okay. So what do you mean by buffer? Okay. So before going in detail about buffer system, how does it will regulate the blood pH, you should know about what is buffer. Okay. Actually, buffer is a substance which will resist the change in the pH even after adding some amount of strong acid or base. Okay. So the substance which can resist the change in pH even after adding small amount of strong acid or base. So that you call it as buffer, and this is a chemically, it is a mixture of weak acid and a salt of its conjugate base. Okay. So here is an example you can consider. So here H2CO3 is a weak acid and its conjugated acid is sodium bicarbonate, NaHCO3. So this is a weak acid and its salt, that is its conjugate base. So this constitutes the buffer. Okay. So ultimately there are various defense mechanisms which will regulate the pH to the normal range. The first line of defense is the buffer system. In the buffer, you should know what is buffer. It will resist the change in pH even after add some amount of an acid or base. This is an example. And suppose if one molecules, so each buffers will differ from another by only a with respect to protons. Otherwise, it is a mixture of weak acid and a salt of its conjugate base. Okay. So here, so what exactly the buffer will do? The buffer is a substance I already mentioned. It will bind reversibly with the hydrogen ions. means whenever it will bind the hydrogen ions, and this is a reaction to show the general reaction is a buffer. So one example is a buffer which bind to H plus ion. This will become this bind to the hydrogen ions. So what does it do exactly? This buffer system doesn't remove the hydrogen ions, or it can neither add into the body, but it only keep them tied up until balance can be re-established. Okay. So previously I told pH is balanced within the reference range. The buffer system which try to maintain the pH either by binding to H plus ions, okay, and it doesn't remove the H plus ions, just bind with the H plus ion, so that it will maintain the pH of the blood. Okay. So we'll go in detail about various buffer system in the body. Okay. So, and also before that, the buffering capacity. Okay. Suppose even if after adding some small amount of an acid or base, so that should pH should be maintained within that range. No, thus buffering capacity depends upon the concentration of a buffer and relationship between pK of the buffer and the desired pH, and also the buffer has a maximum buffering capacity when its pK is equal to pH. This you need to note that. So the buffering capacity mainly depends upon the concentration of the buffer and its relationship between the pK and the desired pH. If pK is equal to pH, the buffering capacity is maximum. Okay. So you consider this, for the maximum blood buffering, the pK of the, that is dissociation constant, that is pK of a buffer should therefore should be near 7.4, so that the buffering capacity will be maximum. So these are the various example for blood buffer system. Okay. So one is, this is a buffer system in the first column. It's a bicarbonate, phosphate and protein. Okay. This buffer system, they may present extracellularly outside the cells. Okay. That is within the blood, and also they may present inside the cells. Okay. That you call it as intracellular buffer. Okay. So various buffer system, that is bicarbonate, phosphate and protein. In bicarbonate, sodium bicarbonate, I already mentioned the buffer is a mixture of an acid, weak acid and its conjugate base. So if you consider the buffer system, the major extracellular buffer is considered to be bicarbonate buffer system. With the similarly, the same component, here the major intracellular ion is potassium. Here the potassium bicarbonate along with carbonic acid. Okay, this is the weak acid and its conjugate base. This is the intracellular buffer. And phosphate as well as protein buffer. Okay, the protein, various protein in the blood, they may exert buffering action. Okay, so this is an example for the various blood buffer. You just need to know about the major extracellular buffer, that is bicarbonate buffer system. Okay, so these are the weak acid and its conjugate base. This is the buffer which is present in the blood as well as inside the cells. That is extracellular buffer as well as intracellular buffer. Okay. Now let us discuss in detail about bicarbonate buffer system. Okay. So sometimes they might ask about the buffer system, or you need to mention about what are buffer, what is the role of buffer. Okay, it will resist the change in pH, and you should give an example various buffer systems. And sometimes they straight away they might ask about bicarbonate buffer system. Okay, so you should know about what is the importance of bicarbonate buffer and how does it will regulate the blood pH to maintain the pH within the range. Okay, so the bicarbonate buffer, it's a mixture of carbonic acid, and here it is NaHCO3. Actually sodium is missing here. Okay, this is the bicarbonate buffer system. The bicarbonate buffer system is the most, I already mentioned, is the major buffer system in the extracellular fluid. Okay, that is outside the cell, and it plays an important role in maintaining the blood pH because of its higher concentration. Okay, because this is present in the blood at higher concentration, so it forms a major buffer system in the blood so that it will maintain the blood pH. What are the two components of the bicarbonate buffer system is, one is carbonic acid and bicarbonate. Okay, so here the two components, the bicarbonate which are regulated by the kidneys and carbonic acids which are regulated by the lungs. Okay, so this is the way the two important components which are present in the bicarbonate buffer system. It is HCO3 minus, that is bicarbonate, and H2CO3 is a carbonic acid. The carbonic acid component which is regulated by the lungs, as well as HCO3 minus concentration, that is bicarbonate which is regulated by the kidneys. So let us discuss how does this bicarbonate buffer will act. Okay, so for any buffer to act, it should resist the change in pH. Okay, so suppose if you add some amount of acid or some amount of base, so you need to know about how does this bicarbonate buffer will react. If you add, you look at this reaction. This is a bicarbonate buffer system. So if you add some amount of acid, so acid will release the H plus ions, the strong acid such as HCl is added to bicarbonate buffer solution. What happens? This H plus ions get released. So the bicarbonate buffer system, it prevents the change in pH. Suppose in the blood, if you add extra increase in H plus ion concentration, that bring down the pH, it lowers the pH. So what does the bicarbonate buffer do? This HCO3 will combine with H plus ion, it forms the H2CO3. Okay, so what is this H2CO3? It is a carbonic acid. Okay, in the next step, it will decompose to release CO2 plus water. Okay, carbon dioxide and water. What does the bicarbonate buffer do? This HCO3 will combine with H plus ions. It forms a carbonic acid. This carbonic acid, it splitted, it decomposed to CO2 and water. The CO2 is diffused to the lung, through the tissues and to the reaches the liver, lungs. From the lungs, it is exhaled out, whereas water is formed, so that it will prevent the change in pH even after adding small amount of an acid. Okay, so this is the reaction which tell about even after adding H plus ions, this will take this H plus ion becoming a carbonic acid, it's splitted and it releases CO2 and water. CO2 is exhaled out through the lungs. Okay, so thereby it maintains the pH. This is one of the mechanisms after adding a strong acid. Then you look at that after adding a base, what happens? So look at this, when sodium hydroxide is a base which is added to the bicarbonate buffer, how does it react? So this hydroxyl ions which is present in sodium hydroxide, that is released from the sodium hydroxide, will combine with, okay, so here look at this reaction so that you can easily understand. The sodium hydroxide, which is in which hydroxyl ions released, and it combined with the sodium which combined with HCO3 and water combined with hydroxyl ion, it will forms the H plus ion combined with hydroxyl ion which forms the water molecule. Okay, so even after adding an base added to the bicarbonate buffer, the hydroxyl ions from NaOH combines with H2CO3 like this, NaOH, H2CO3, it forms sodium bicarbonate with the release of water molecule. So what you understand from this reaction is, even the base is added, this bicarbonate buffer system has a two component, one is H2CO3, another one is HCO3 minus. Here it will accept these hydroxyl ions, okay, it combined with H plus ion, it forms a water molecule, and sodium combined with HCO3, it will form sodium bicarbonate. Okay, this is a summary of the reaction. How does the blood buffer, especially the bicarbonate buffer, will react even after adding strong acid or base. Okay. Now, so in detail about the bicarbonate buffer system. So any non-volatile acid which is stronger than carbonic acid can be buffered by bicarbonate. Okay, so this buffering action of the bicarbonate is mainly which is carried out to any non-volatile acid, okay, that is stronger than the carbonic acid, which are buffered by the bicarbonate. And this bicarbonate is a measure of the base, okay, and that remains even after adding small amount of an acid which is stronger than the carbonic acid, which are neutralized by this bicarbonate buffer system. One thing you should know that the bicarbonate buffer system, where the bicarbonate level which forms the reserve of alkali which is available for neutralization of the strong acid, okay, this you call it as alkali reserve, okay, this you call it as alkali reserve. And you need to remember this thing, this might be asked in MDMS entrance question, what is the alkali reserve of the bicarbonate buffer system? Look at this, there are two components which are present in bicarbonate buffer, that is HCO3 and H2CO3. Here HCO3 minus, this is the alkali reserve, that is plasma is about 25 millimoles per liter, okay, to H2CO3 concentration is just 1.25 millimoles per liter. So the proportion is about 20:1. This is the proportion of alkali which is reserved, so that bicarbonate buffer has the largest buffering capacity, so that it has more of alkali in nature. It has contains 25 millimoles per liter when compared to H2CO3 concentration, that is carbonic acid concentration, okay. So you should remember this 20:1 ratio of the HCO3 to the H2CO3 concentration, okay. So if any changes in the concentration of either HCO3 or H2CO3, therefore in the ratio of which if it is alters, that bring about a change in pH, okay. So this is the pH which is maintained within the range is mainly because of it is the alkali reserve, that is 25 millimoles per liter to the 1.25 millimoles per liter of carbonic acid, that is 20:1. If this ratio alters, then definitely it will change the pH of the blood. So ultimately in bicarbonate buffer system, the two elements of buffer system, that is HCO3 minus and H2CO3, so which are regulated by, so look at this, this HCO3 and H2CO3 which are regulated by the kidneys as well as respiratory regulation. In the kidney, the bicarbonate concentration which are regulated either by increasing or decreasing the rate of reabsorption of bicarbonate by the kidneys, and in the lungs by altering the rates of removal or retention of carbonic acid by the lungs, okay. This is the two way in which bicarbonate buffer system which will maintain the pH. One is by through the kidney, either by increasing or decreasing the rate of reabsorption of HCO3. Second one is by altering the rates of removal or retention of H2CO3 by the lungs, okay. Now the phosphate buffer system. So previously I discussed on bicarbonate buffer system. The bicarbonate buffer system can be separately asked as a 5 marks question or 3 marks question. What you are supposed to write is, you should define the buffer and its role and just elaborate the points which favors about bicarbonate buffer system and its what are the components in bicarbonate buffer system. How does bicarbonate buffers which will regulate the pH even after adding small amount of an acid, what is the change which occurs even after adding small amount of base in the form of hydroxyl ions, what is the change will occurs, the reaction you can mention, and also these two elements which are maintained by the kidney and lungs, you need to highlight about the bicarbonate buffer system, okay. Now the phosphate buffer system, okay. So that is this is also one of the buffer system which is present in the blood. The phosphate buffer system is not so important as compared to blood buffer, because this phosphate buffer compared to bicarbonate buffer, it's a relatively minor component. But this plays a major role in buffering renal tubular fluid and intracellular fluid. Okay, so the phosphate buffer plays a major role, especially in case of renal tubular fluid handling and intracellular fluids. So that is the role of phosphate buffer system. Okay, how does the phosphate buffer system will act? So for any buffer which resists the change in pH even after adding an acid or alkali, we need to mention how does it act. That is mechanism of action of phosphate buffer. So suppose when a strong acid such as HCl is added to the phosphate buffer, what does it do? Okay, so here the H plus ions is accepted by the base. The base component is, so here phosphate will accept the H plus ion. This will become dihydrogen phosphate. Okay, so hydrogen phosphate become dihydrogen phosphate, and this strong acid HCl, which is replaced by the weak acid, that is so sodium dihydrogen phosphate. Okay, this is a change which occurs even after adding small amount of an acid. Okay, strong acid such as HCl is added to the phosphate buffer. Okay, so this is what you should write even after adding an acid. What is the change occur? How does it doesn't change the pH by combining with the protons, so that it doesn't alter the pH of the blood. Okay. So now this is a reaction. If HCl, look at this. Here the base component, so disodium hydrogen phosphate. So HCl, it releases a proton, so that is accepted by the disodium hydrogen phosphate. It become sodium dihydrogen phosphate. Okay. So and the sodium chloride get released. Okay. This is hydrogen dihydrogen phosphate is formed. That is the thing you should understand. Okay. At the same time, when a strong base such as sodium hydroxide is added to the phosphate buffer, what happens? So this is hydroxyl group is buffered by dihydrogen phosphate to form HPO4 2 minus, okay, and water molecule. So look at this reaction so that you can easily know. Sodium hydroxide combined with sodium dihydrogen phosphate. Okay, so here the base you are adding. So this will accept the base of the phosphate buffer, so that it will become disodium hydrogen phosphate with the release of water molecule. Okay, this is how the phosphate buffer accept the proton as well as the base in the form of hydroxyl ions, so that it will doesn't alter the pH of the blood. Okay. So at plasma pH of 7.4, what is the ratio of this? HPO4 2 minus and dihydrogen phosphate. It is the ratio is 4:1. Okay, that is 4:1. Okay. This is about phosphate buffer. Even after adding an acid or alkali, what is the change which occurs? Okay. And there is one example. Okay. Just wait a minute. There is some disturbance. Okay, sorry for the inconvenience. There is some construction work is going on. Okay, so you might be having some interruption. So I was discussing the phosphate buffer system. Here, I told in previous slide, even after adding small acid or alkali, this will accept the proton as well as the base, so that it doesn't change the pH of the blood. So one of the example, the organic phosphate which is formed, okay, that is 2,3-bisphosphoglycerate in the RBCs, that forms the non-carbonate buffer system of the RBCs. Okay, so that is also one of the organic phosphate buffer which is present in the RBCs. It forms 16% of the accounts for the 16% in case of red blood cells. Okay, this is an example you can quote for this phosphate buffer system. Okay. So now, I discussed the bicarbonate buffer system. This is a major buffer system in the extracellular fluid. In the phosphate buffer system, so especially in case of renal tubular fluid handling, that forms an important thing. And now it is a protein buffer. Okay. So here the various proteins in the blood which may affect, which exhibits the buffering action. In that mainly the protein, mainly the albumin which exhibit the buffering action, and also I discussed about the hemoglobin which acts as a buffer. So these two things I am just going to highlight about protein buffers. So here this protein which can bind to the base as well as protons, okay, in the form of sodium protein or H plus protein. Okay, so that's why it acts as a conjugate weak acid and its conjugate salt. Okay, so which will bring about mainly the buffering action, mainly the content of protein. The proteins are made up of amino acids. The amino acids, it may be acidic or basic amino acids. So acidic amino acid may accept the hydroxyl ions, whereas basic amino acid may accept the H plus ions. So in that way, it will maintain the buffers. So just to mention, in that proteins, mainly plasma protein, albumin acts as a major buffer. Okay. In acid solution, the basic amino groups, suppose in a acidic medium, the H plus ions are high. The basic amino groups, I mentioned that takes up this excess H plus ions and forms the NH3 plus, so that it will maintain the blood pH. So any buffer, if you are going to explain any buffer system, so you need to mention the components and what happens after adding small amount of acid, how does it react, and what happens after adding small amount of base, how does it react. So you need to mention about that. Okay. So here, if the base solution like COH groups which is added, so this basic solution in which the content of acidic amino acid which will accept the hydroxyl ions from the bases which are present in a solution. So what does it do? It forms a hydroxyl ion combines with this COH group which is present in amino acids. That means to say in the proteins, that forms a water. Okay. So what you take home message is, a buffer is a solution which resists any change in pH. It will accept the proton, okay, as well as the base. So that because the proton is accepted by the salt content, that is basic component of the buffer, as well as hydroxyl ions which is accepted by the weak acid component, so that it doesn't allow any change in pH. You need to explain this. Okay. So other important things which in the proteins which will control the blood pH is mainly because of the histidine is an amino acid which contain imidazole groups. Okay, so that is also will maintain the blood pH even at physiological pH, that is maintained because of the content of the histidine residues. Okay, and also the albumin contains about 16 histidine residues. Okay, so that forms a major buffering role of a protein buffer. So this might be asked in MDMS entrance question. Among the protein buffer, which is the amino acid which mainly contribute in maintaining the blood pH? Okay, that is the histidine residues. So this imidazole group of histidine is mainly responsible for that. So this is the one which will maintain the blood pH. Okay, and hemoglobin buffer. Okay, so look at this. The hemoglobin can combine with potassium ions as well as H plus ions. The main function of hemoglobin, it carries the oxygen from lungs to the tissues, and also simultaneously it carries the carbon dioxide, so from tissues to the lungs. In which form I will discuss that so that you can understand clearly how does it will act as a buffer. Okay, let us see. This is a hemoglobin forms a major intracellular buffer of the blood. Okay, this is mainly present in the red blood cells. Okay, so this it's a intracellular buffer system, and each hemoglobin can contains it contains 38 molecules of histidine. So similarly, I mentioned for albumin, it contains 16 histidine residues, whereas hemoglobin contains 38 histidine residues. So this is an amino acid which will maintain the blood pH in the physiological range because of this presence of the histidine residues. Okay, and because the imidazole group of histidine has a pK approximately 7.3. That's why histidine is a major component which determine the which will maintain the blood pH, because its pK is equal to the pH of the average pH of a solution. Okay, that is the reason imidazole group of the histidine residues which forms a which contributes to the buffering action. Okay, and this hemoglobin buffers which buffers the carbonic acid. Okay, so look at this. How does it bring about these changes? Mainly, the hemoglobin works effectively in cooperation with the bicarbonate buffer system. Okay, so look at the diagram. This is the lungs in the left hand side. In the middle one is the blood. Okay, what are the various things which are diffused through the blood? In the right hand side, so to yours right, it's a tissue. It contains the RBCs. This is the red blood cells. You start from here so that you can easily understand. During various tissue metabolism, CO2 is released, carbon dioxide is released. Okay, what is the fate of carbon dioxide? It will combine with water molecule. It forms the carbonic acid. Okay, this carbonic acid, it is decomposed to H plus and HCO3 minus. Okay, here H plus ions are increased in intracellularly. So just now I mentioned this one of the intracellular buffer, that is hemoglobin buffer, which will maintain the blood pH. How does it maintain? This H plus ions, which is taken up by the hemoglobin, that is oxygenated hemoglobin, there it will release, it will exchange the H plus ion and release the oxygen. So that's what you call it as Bohr effect. You might have studied in physiology in detail. Okay, so ultimately the hemoglobin, the oxygenated hemoglobin accept the H plus ions, oxygen get released, and this forms the proton is accepted by hemoglobin. This will become HHB. Okay, the protonated hemoglobin. Okay, this is easily diffused through the blood, and this will reaches the lung. Okay, so what happens in the lung? Okay, this has a more affinity towards the oxygen, because higher concentration of oxygen it combines with HB and with the release of this proton H plus ions, so that again it becomes oxygenated hemoglobin. Okay, this is freely diffused across the lungs and membrane, and through the blood it again reaches the red blood cells. So in this way, this is a cycle which continuously it will be operating, where the carbon dioxide enters in the tissue, it forms carbonic acid. H plus ion is accepted by the oxygenated hemoglobin. Okay, and oxygen get released. Again, it diffuses through the blood and reaches the lungs, where it combine with one more molecule of oxygen, so that hemoglobin will carry oxygen from lung to the tissues and get released, and simultaneously it carries the proton and get released to the lungs. Okay, So then what is the fate of these protons? These protons released, it combines with bicarbonate, that is HCO3, that will form carbonic acid, that is H2CO3, and it will split up to carbon dioxide and water. Okay, this combined with carbonic acid, this will split up to form carbon dioxide and water, and this will be it will be exhaled. The CO2 is exhaled. So where do you get this bicarbonate? This bicarbonate or alkali resin you got from the RBCs and various metabolic process. We call it as chloride shift. This is an anion transporter which is present on the lungs, where it will exchange the bicarbonate with the chloride ions. Chloride ions are moved out and bicarbonate are entered inside. Okay. So this is what is a summary of hemoglobin buffer. It will take up the protons, and this is the way it will be removed in the form of CO2 and water molecules through the expiry. Okay, it's about the hemoglobin buffer system. So the same thing I explained in words. The transport of this CO2 which is released from tissue without any change in pH, we call it as isohydric transport. And most of the CO2 which is transported in the plasma, in which form? Just now we have studied, the form of bicarbonate. So if bicarbonate is much more soluble in blood than CO2, this cause increase in blood capacity to carry more CO2 from tissue to the lungs. Okay. Ultimately, more and more bicarbonate, more and more the CO2 get released, and CO2 is effectively reutilized, and this will bring about the change so that it maintain the blood pH. Okay. So that is about how does the hemoglobin buffer which maintains the blood pH. What do you mean by respiratory mechanism? Okay. So in respiratory mechanism, it's the second line of defense against acid-base disturbance. What is first line of defense? Just now I discussed about buffer system, that is bicarbonate buffer system, and protein buffers, phosphate buffer, as well as this hemoglobin buffer. Okay. This respiratory mechanism, it acts by regulating the concentration of carbonic acid. This is very important. So this is what I am discussing. Today's topic is if you need to understand each and every line so that you can apply clearly. Otherwise, so you should understand the concept and you can apply. Okay. Here is the respiratory mechanism to regulate the pH by changing the concentration of carbonic acid in the blood and other body fluids by the lungs. Okay, that is by respiratory regulation. How does it do? This respiratory center, which will be under regulation with the level of CO2 concentration. Okay, so what does it do? This respiratory center, either they will remove the CO2 or will retain the CO2. Thereby, H2CO3 concentration will change so that it will maintain the pH of the blood. Okay. How does it bring? So look at this. When small amount of H plus cyanide added, okay, or H2CO3, this will stimulate the respiratory center. When the protons are added, this will stimulate the respiratory center, and the rate of respiration will be increased, and excess acid in the form of CO2 is quickly removed. This is the way in which the respiratory system, which will regulate the blood pH even after adding small acid. Suppose if you add small amount of base in the form of hydroxyl ions or bicarbonate. Okay. So what does it do? It will depress the respiratory center so that the ventilation will be decreased, and the release of CO2 from the blood also decrease. So that CO2 is retained, so that even after adding an alkali, the pH start increasing, because CO2 is retained. Combined to water, it will form carbonic acid. Okay. Even after adding base, the acid is formed here with this mechanism. So here, after adding an acid, this forms a base so that it will maintain the pH. Okay. So This is a summary. The increased blood CO2 will result in the formation of H2CO3. Okay, this will neutralize the excess alkali. This is about respiratory regulation. The renal regulation I will be discussing in the next class in detail. Just to show some introduction about renal mechanism. This is the third line of defense. First line of defense is buffer system. Second line of defense is respiratory regulation. Third line of defense is renal regulation. So here, this is exerted by the various mechanism, that is mainly by conserving the HCO3 concentration. It will maintain the alkali reserve or by excreting the acids. Okay. So by two mechanisms, the renal system will maintain the blood pH. Okay. So in next class, I will be discussing in detail about renal mechanism in acid-base balance. Okay. Summary, in today's class summary, I taken that what is the normal pH of arterial blood? So it's about 7.35 to 7.45. Some books mention 7.36 to 7.44, and the average is 7.4. Okay, it's a neutral pH. And how does it is maintained within that range with the help of three line of defense, and this is by buffer system as well as respiratory regulation and renal regulation. I discussed on different buffer system in the body as well as respiratory regulation. In the different buffer system, you need to learn about bicarbonate buffer system. That's very important. How does it react even after adding small amount of acid or alkali? How does it maintain? And also about hemoglobin that you have learnt in the physiology, but how does it maintain? You just learn about that. And this respiratory regulation. So how does it will maintain the blood pH after adding a small amount of an acid or alkali? Okay. This is about today's class. In the next class, I will just discuss on renal regulation and disorders of acid-base balance, about acidosis and alkalosis and mixed acid-base disorder. Okay. Thank you. Any doubt? You just ask in the group, so that will help you. All of you just start preparing about whatever the classes which is covered for your internals, that will be asked all these topics.